Imagine you're sitting in a quiet room, thinking about how we actually know what we know about the human mind. Every fact you've learned in psychology, how memory works, why people behave the way they do, what helps us learn better, all of it came from someone asking questions and carefully searching for answers. This is the heart of research methods, the tools psychologists use to understand human behavior and thought. Sometimes, the richest understanding comes from following one person's journey in incredible detail. Think of it like writing a biography, but for science. When researchers do this, focusing deeply on a single person, family, or even an organization, they're conducting what we call a case study. The beauty of a case study is in its depth. Because you're not spreading your attention across hundreds of people, you can notice the small details, the patterns that emerge over time, the unique story that unfolds. This gives your findings real richness and validity. They feel true because they capture real life as it actually happens. You're watching someone in their everyday world, which means the study has high ecological validity. It reflects real life, not some artificial situation. But here's the thing about unique stories: they're unique. When you study one person so closely, you learn everything about that person, but it becomes harder to say whether what you found would apply to others. Scientists call this difficulty with generalization, taking what you learned in one situation and applying it more broadly. Now imagine you're curious about how children interact at recess, or how people behave in a coffee shop. You could just watch. This is what psychologists call observation, and it's more nuanced than it might seem. When you're observing, you have choices to make. Will the people know you're watching them? If they do, you're being what researchers call an overt observer. You're open about it. If they don't know, you're a covert observer, perhaps blending into the crowd, watching quietly from within. You also need to decide, will you be part of the action or will you stay on the sidelines? A participant observer joins in, experiencing the situation from the inside, maybe joining that playground game or sitting at that coffee shop as a regular customer. A non-participant observer stays removed, watching from a distance, not getting involved. The way you watch matters too. Sometimes researchers use what's called a structured observation, where they have a specific checklist of behaviors they're counting, like tallying how many times children share toys. Other times, they go broader with an unstructured observation, taking in everything, usually when they're first exploring a topic and aren't sure what to look for yet. And then there's the setting. A naturalistic observation happens in the real world, that actual playground, that real coffee shop, where everything unfolds as it naturally would. But sometimes researchers set up situations on purpose, carefully arranging things to see what happens. That's a controlled observation, where the environment has been shaped by the researcher. Each approach has its trade-offs. When people don't know they're being watched, they act naturally, giving you that valuable ecological validity. But if they do know, they might change their behavior, showing what they think you want to see, more socially desirable behavior rather than their genuine responses. This can reduce the validity of what you find. There's also the matter of ethics. When you're watching people who don't know they're being studied, you can't really ask for their informed consent, their agreement to participate based on understanding what's happening. That raises important ethical questions that researchers must carefully consider. Sometimes in life, you notice patterns. Maybe you notice that on days when you drink more coffee, you feel more anxious, or that people who exercise more seem to sleep better. These relationships between things are what psychologists study through correlations. A correlation is simply a relationship between two variables, two things that can change. But here's what makes it different from an experiment. You're not changing anything on purpose. You're just measuring both things and seeing if they move together. When both things increase together, like exercise and sleep quality both going up, that's called a positive correlation. When one goes up while the other goes down, like caffeine and calmness perhaps, that's a negative correlation. And sometimes they just don't seem related at all. That's no correlation. The crucial thing to remember, and researchers emphasize this constantly, correlation doesn't mean causation. Just because two things move together doesn't mean one is causing the other. There might be a third factor at play, something else influencing both variables that you haven't noticed yet. Before any study begins, researchers need clarity about what they're looking for. This starts with an aim, a simple statement of what the study intends to explore or show. It's broad, like to investigate whether light levels affect concentration. But to really test something, you need to be more specific. You need a hypothesis, a testable prediction that you can actually measure and either support or disprove. This is sometimes called the alternative hypothesis because it proposes an alternative to the idea that nothing special is happening. Hypotheses come in flavors. A directional or one-tailed hypothesis makes a specific prediction about which way things will go. People will concentrate better in bright light than in dim light. A non-directional or two-tailed hypothesis just predicts that there will be a difference, without saying which direction. There will be a difference in concentration between bright and dim light. And then there's the null hypothesis, which basically says nothing interesting is happening here. Any differences we see are just random chance. Scientists keep this in their back pocket to test against their main prediction. In experiments, everything revolves around variables, things that can change or vary. The most important distinction is between two types. The independent variable, or IV, is what the researcher deliberately changes or manipulates. Think of it as the thing you're testing, maybe different light levels or different amounts of sleep, or whether someone has eaten breakfast. The researcher creates different conditions of this variable to compare them. The dependent variable, or DV, is what you measure to see if the IV had an effect. It's called dependent because it depends on what happened with the independent variable. So if your IV is light level, your DV might be a score on a concentration test. But here's where it gets tricky. There are other variables lurking around that could mess up your results. Things like whether people are tired or hungry or just came from a stressful class. These are extraneous variables, and they need to be controlled, kept the same for everyone, so they don't confuse your results. When an extraneous variable does sneak in and affect your results, particularly if it affects only one condition, it becomes a confounding variable, and that's a real problem. To make sure everyone knows exactly what you're measuring, variables need to be operationalized, defined in precise, measurable terms. Happiness is vague. Score out of 10 on a happiness scale is operationalized. Once you know what you're testing, you need to decide how to arrange your participants across the different conditions of your study. This is your experimental design, and there are three main approaches. In an independent measures design, different people do different conditions. Group A experiences one condition, Group B experiences another, and you compare the groups. It's called independent because the data from each group is independent, it comes from different people. The advantage: participants only see one condition, so they're less likely to guess what you're studying. The challenge: people are different from each other, and these participant variables, individual differences like personality, intelligence, or mood, might affect your results. To help with this, researchers use random allocation, putting people into groups by chance so that differences between people get spread evenly across conditions. In a repeated measures design, the same people do all the conditions. Everyone experiences everything. This is brilliant for controlling participant variables. Each person is being compared to themselves, so individual differences don't matter as much. But now you have a new problem: order effects. When people do something twice, they might get better at it the second time, a practice effect, or worse because they're tired or bored, a fatigue effect. To handle this, researchers use counterbalancing. Half the participants do condition A then B, while the other half do B then A. This is sometimes called an ABA design. The third approach tries to get the best of both worlds. A matched pair's design takes different people, but carefully matches them in pairs based on characteristics relevant to the study, maybe age or intelligence or initial skill level. Then one person from each pair goes into each condition. It's clever but challenging, because finding people who match well takes time and effort. Experiments themselves come in different forms, depending on where and how they're conducted. A laboratory experiment is what you might picture when you think science. A controlled setting, usually not the participants' normal environment, with careful controls over everything. The room temperature is set, the lighting is precise, the procedure is exactly the same for everyone. This standardization, keeping everything exactly the same, helps with reliability, meaning the study is consistent and could be repeated with similar results. But that artificial environment can be a double-edged sword. Yes, you have great control, which improves your validity, your confidence that you're really testing what you claim to be testing. But participants might not behave naturally in a lab. They might feel nervous or try to guess what you want, responding to demand characteristics, cues in the study that give away its purpose. A field experiment takes the experiment out into the real world. You still have an IV and DV, you're still manipulating something on purpose, but now it's happening in the participant's natural environment, maybe their actual classroom or workplace or home. The behavior feels more real, more generalizable to other situations, with better ecological validity. But you lose some control. There are more uncontrolled variables floating around that might affect your results. A natural experiment is a bit different. Technically, it's not a true experiment at all. Here, the researcher can't actually manipulate the IV. Instead, they're studying something that's already different or has changed naturally. Maybe comparing children who grow up with TV to those who don't, or looking at behavior before and after a new law is passed. You can study important real-world questions, sometimes even things that would be unethical to manipulate on purpose. But with less control comes less certainty about what's causing what. Every study needs participants, but you can't usually study everyone you're interested in. The entire group you're interested in is called the population. The subset you actually study is your sample. How you get that sample matters, and there are different sampling techniques. Opportunity sampling is the simplest. You just use whoever is available. Students in the cafeteria, people walking past on the street, whoever you can get. It's quick and easy, but your sample might not represent the broader population very well. Volunteer or self-selected sampling is when you advertise and people come to you. They might see a poster or email and sign up. These participants are likely to stick around since they chose to participate, but they might be a specific type of person, the kind who volunteers for studies, which limits generalizability. Random sampling is the gold standard. Every person in the population has an equal chance of being selected, maybe by picking numbers from a hat or using a random number generator. In theory, this should give you a representative sample. In practice, it can be hard to pull off, and even with random selection, some people still won't participate. Two questions haunt every researcher. Is this real, and is this consistent? Validity is about truth. Are you actually measuring what you think you're measuring? Internal validity asks whether your study is well controlled enough that you can trust the relationships you found. Ecological validity asks whether your findings would apply in the real world, beyond your study. Face validity is simpler. Does your measure look like it's testing what it claims to? And concurrent validity checks whether your measure matches up with other established measures of the same thing. Reliability is about consistency. Internal reliability means your procedure is standardized enough that each participant gets the same experience. External reliability means that if you did the study again, you'd get similar results. Inter-rater reliability is about whether different researchers would score or interpret the same data in the same way. To test reliability, researchers might use the test-retest method, giving the same test twice and seeing if people score similarly both times. Or the split-half method, checking if the first half of a questionnaire gives similar results to the second half. Once you've collected your data, you need to make sense of it. Numbers on their own don't tell you much. You need to summarize and describe them. Measures of central tendency tell you about the average or typical score. The mean is what most people think of as the average. Add everything up and divide by how many scores you have. The median is the middle score when you line everything up in order. The mode is the most common score, the one that appears most often. Measures of spread tell you how varied or spread out your data is. The range is simple, the gap between your highest and lowest scores plus one technically. Standard deviation is more sophisticated. It tells you, on average, how far each score is from the mean. Sometimes data follows a normal distribution, that beautiful bell-shaped curve where most scores cluster around the middle and fewer trail off on either side. The mean, median, and mode all sit together at the peak. To visualize your data, you might use bar charts for separate categories, histograms for continuous data that flows along a scale, or scatter graphs to show how two variables relate to each other in a correlation. Sometimes you need to ask people directly about their thoughts, feelings, or experiences. This is where self-reports come in, questionnaires and interviews. Questionnaires are written questions, often completed privately. They might use Likert scales, which are agree or disagree scales, rating scales where you rate from 1 to 10, closed questions where you choose from set answers, or open questions where you write whatever you want. They're efficient. Lots of people can complete them quickly, and participants might be more honest when they're not face-to-face with someone. But people might still give socially desirable answers, saying what sounds good rather than what's true. Interviews involve direct conversation. Structured interviews follow a fixed script, the same for everyone, highly standardized. Semi-structured interviews have set questions, but allow the interviewer to dig deeper with follow-up questions. Unstructured interviews are more like guided conversations, following wherever the participant's answers lead. Interviews can gather rich detail, but participants might feel less comfortable being honest face-to-face. Throughout all of this, every study, every method runs a crucial thread: ethics. Participants deserve respect and protection. They should give informed consent, knowing what they're agreeing to. They should have the right to withdraw at any time. Their privacy shouldn't be invaded, and their confidentiality should be protected. Sometimes research creates tension between good ethics and good science. The most natural behavior might come from participants who don't know they're being studied, but that raises ethical questions about consent. Researchers must constantly balance these concerns, ensuring that the pursuit of knowledge doesn't come at the cost of people's dignity and well-being. And so, these are the tools, the methods and concepts that psychologists use to explore the human mind. Each has its place, its strengths, its limitations. Together, they form a toolkit for understanding ourselves and others, for testing ideas carefully, for building knowledge that we can trust. As you drift off, let these concepts settle in. They're not just abstract terms, but ways of thinking, ways of knowing, ways of seeking truth about what it means to be human.